This is our session 3 of our nCounter Gene Expression Masterclass. We've already done 2 sessions on nCounter Gene Expression data analysis. If you're curious about those, you can find them on our website. I'll paste the blog link here. We have done recaps of those, so in a blog format, written format, or there's also video recordings if you prefer to see it that way. We're very excited to have Ryan here. Ryan is our in-house NanoString expert. He does a great job explaining the complex of topics in the most simplest of ways. So we're really excited on today's topic, which is quality control. As a former bioinformatician, I know that quality control is super important for data analysis, and it's where you basically make the decisions of which samples you want to include, which samples you want to exclude, that kind of thing. Sometimes it's also tricky to understand what metrics you want to use for those. So I'm very excited to listen to this session on nCounter today. So yeah, without further ado, I'll let you take the floor, Ryan.
Thank you, Cookie. I appreciate that introduction. So I am excited to be continuing in our Masterclass series here. Today we're focused on quality control. So there's going to be a heavy focus on looking at individual metrics, how they're calculated, how they're interpreted at a high level conceptually. So what you'll be seeing is a lot of very detailed slides. The slides are going to be more detailed than they typically would be if I was just doing a regular webinar or seminar. And these are meant to be used as material for you for future reference. So if you're not able to take all of the notes, write down all of the details, I put it down on the slides for you. So I might suggest that you maybe just focus on listening and thinking about what I'm saying rather than trying to write down every detail. But just know the slides will be available after the fact. So if you're not able to write down anything, that is fine. So let me get started. I'm going to quickly give a brief background on myself here. So what's relevant for today's session is that I have about 10 years of experience working with NanoString nCounter data. So I'm a PhD bioengineer by training, so I've been heavily focused on genetics and gene expression, genomics for my entire career, and I'm quite interested in drug development. But I did work at NanoString for about 7 years as the primary leader of the FAS team on the West Coast. And I've worked with pharma institutions, biotechs, as well as academics with a wide variety of sample types and experimental designs. So I have a lot of expertise in the chemistry, the instrumentation, but of course also the data, what we're talking about today. So this presentation is going to be heavily focused on slides for the next 20, 30 minutes, and then we're going to jump into Rosalind towards the end. But let me set the stage. I'm seeing a lot of new names on today's broadcast, so welcome everyone that is here for the first time for this session, and thank you all that are returning as well. I'm glad you're finding these sessions useful. To set the stage, so at a high level, why I got started in science is that I wanted to make a difference. I wanted to improve healthcare, well-being, and health outcomes for everyone. I wanted to challenge and identify what are the root causes of diseases that were affecting the population, such as cardiovascular disease, and use my knowledge of genetics to potentially develop new therapeutics. And that perfectly aligns with the mission of Rosalind in general. So we developed this unified multi-omics platform to enable all of you to analyze your data and make those great scientific discoveries, which push human health and well-being forward. So the purpose of this masterclass was shown here as 4 main things. The first is that I wanted to solidify the value for you, the synergy of pairing Rosalind and nCounter technology together. Each on their own as they stand, they're great technologies, but paired together is when you truly unlock the value of each of them. So throughout this masterclass, I want you to gain a deep understanding of the algorithms. How does normalization work? How does pruning work? And in this case, how does QC work? Because it's in my experience, when you truly understand how the metrics are calculated, that's when you really understand what you're doing and gives you confidence to making sure you're making the correct decisions in QC. So it's all about giving you confidence and enabling you to make better decisions as you analyze your data. So today's session, we're focused on quality control. If you missed the previous sessions, those were on custom pruning and custom normalization. We do have recordings available. So if you want information on how to find those, please reach out to us. You can also go to Rosalind Academy to find them there. I'm not going to do a big introduction to nCounter technology. I think everyone here appreciates the beauty of the nCounter technology. It's time to insight, its speed, how easy it is to perform in the lab, and how minimal technical variation and technical bias there is in the data. It's just a fantastic technology. Been around, I think, almost 20 years now. So I love using the technology and teaching about it because it's so powerful and elegant. But what's relevant today, of course, is the data itself. So I visualize this as an abacus on the right-hand side, just kind of as in jest, just signifying that the data they're simple, they're intuitive, they're easy to analyze and understand. So this sets NanoString nCounter data apart, is that it's really being able to analyze by the end user, by the scientist. You don't need that bioinformatics core necessarily to do it for you. So we'll be seeing various parts of the Rosalind platform as we progress through this masterclass. Today we'll be focusing on the quality control module within the Rosalind platform. There's various other modules which we may or may not discuss over the course of the next month or so. But today we're focused on quality control, and upcoming we'll see some of our pathway features and cell profiling features and meta-analysis. What the origin of this masterclass was is that we wanted to launch this in conjunction with all of our new nCounter features. So we had some new custom pruning capabilities, custom normalization capabilities. We enhanced the pathway features, our intelligence and knowledge and meta-analysis and cell profiling. So we wanted to launch this masterclass educational series in conjunction with this to ensure you can get the best data analysis possible. And in my experience working with data, with nCounter, with researchers for over the past 10 years, Rosalind is the place to analyze your nCounter data. It's the most powerful and collaborative solution at the institutional level to analyze your nCounter data. So before I skip into talking about some QC, I just want to finish with this summary slide here. And again, I just want to reiterate that the combination of Rosalind and nCounter together is where you truly unlock the value of the technologies. So this diagram down here at the bottom represents the workflow. Starting in the lab as the bench scientist, you're setting up your hybridization in 15 minutes of pipetting. You load that into your instrumentation that processes overnight in your thermocycler. Then you load it into the nCounter instrumentation, and you have your data from sample to raw data in less than 24 hours. And at that point, you load it directly into the Rosalind platform, and then you can unlock all of the enhanced intelligence capabilities, knowledge capabilities that are available in the platform. So together is when you truly unlock the value of the two technologies. Okay, so that being said, let's start talking about quality control. So I'm going to take a second here, like I do in all of the previous classes I've had here, and pose a question to you to think about quietly for a few seconds here. Why do we want to perform quality control of our nCounter data? So think about that for a few seconds, and then I'm going to share my thoughts, but I just want to force you all to get back to basics and to start from the beginning. Why do we do this? Why is it important? Okay, so hopefully a few thoughts popped into your mind. I will show you what first came to mind when I was thinking about this question. Two things. The first is that you want to assess the suitability of your samples for inclusion or exclusion from an experiment. And why do we want to exclude certain samples? It's because we want to identify these quote unquote bad samples, get them out of the data set, get them out of the analysis, because if they're included and we include bad data in your analysis, we might have spurious biological interpretations, you might have incorrect results. So it's all about looking at our samples in depth with a series of QC steps to figure out which samples should be excluded so that we have the best outcome, the best interpretation possible. That's quality control in a nutshell, at least from my perspective. So how has quality control been done traditionally in nCounter for gene expression data? So on the left-hand side, I show you kind of the two standards that have been in place for about the past 15 or 20 years or so, and that's nSolver. Up at the top, nSolver Basic, it's that local software you install on your local computer. That established some standard best practice metrics that everyone's been looking at for the past 20 years. Imaging, QC, positive control linearity, limit of detection, so these and some additional metrics as well. So those are the standard metrics that have been around for a while. Below that is the box for nSolver Advanced Analysis, which was released a little later than the basic version. and that introduced some visualizations. They were limited in scope, they were static images, and there was actually a decrease at this point in the number of metrics that were available. If you're not aware, advanced analysis is an R plugin into nSolver Basic. So something that happens is that you get some nice QC metrics in nSolver Basic, but then when you generate the results in nSolver Advanced Analysis, you don't get a reproduction of all those QC metrics. So your QC becomes fragmented. Part of it is left in nSolver Basic, and part of it is found only in Advanced Analysis. So if you want trying to share that data with any colleagues, there's a very high risk that you don't transfer all of the QC information to your colleague. So using a solution like Rosalind and a cloud-based platform is essential for collaboration. So all of your data is found in one place, nothing becomes fragmented. So in addition to incorporating all of the metrics and QC features from basic and advanced analysis into Rosalind, we also formed a partnership with NanoString, which some of you might know. So we were thinking about how can we enhance the QC experience for nCounter data in the Rosalind platform. The Rosalind team, the data science team, bioinformatics team, we had some ideas and we incorporated them into Rosalind. And then NanoString, as part of our partnership, they suggested some additional QC features that they wanted to see included within Rosalind, so we added those as well. So going forward, all of the new nCounter analysis features will be found within the Rosalind platform. So from my perspective, it's clearly the best place to be QCing your nCounter data. So that being said, let's pretend you uploaded your QC data into the Rosalind platform and you generate results and you start looking at your QC. You might see something like this. If you're not familiar with QC, you just get this information that comes up, and if you don't know how to interpret it, it might look kind of like gibberish like this. You have all these different QC metrics, they're all associated with numbers, and we need to find some sort of structure and framework in order to interpret this. If you're seeing this for the first time and you haven't had training on how to navigate this information, it's going to be pretty dicey. So here and now, I want to present to you a framework to enable you to navigate through this with confidence, with ease, understand what you're doing so you get those outcomes that you want. So we can go from this and then we can rearrange things, and we do this within the Rosalind platform. But we can go from this, just kind of this bolus of information, I'll call it, and we can start to organize it and put it into some context here. So let's go from here to this next stage, which is organizing the metrics like this. So on the left-hand side, we have what I'm calling metrics. So a metric is just a number or a score, like 0.5, 1.7, some number that you're assigning to a sample. So these are metrics for individual samples. So if you have 12 samples in your data set and we're looking at that imaging quality metric, you'll have an individual imaging quality metric for all 12 samples. So these metrics, they're numbers, they're assigned for each of these metrics to all samples individually in a data set. That's on the left-hand side. On the right-hand side, now we can start talking about visualizations. Typically, visualizations take all of the data, or takes data from all of the samples to visualize and help you interpret at the same time. So it's more of a consolidation of all samples in your data set for visual representation and interpretation. So again, it's individual metrics on the left versus this more consolidated approach with visualizations on the right. But within each of these boxes, we have two different types of QC information, and I split them into technical versus biological. Technical metrics or technical visualizations are showing us some information about the technical performance of the assay. So it's really, how well did the hybridization work? How well was the imaging performed? How well did the instrumentation work? Just technical parameters associated with the assay itself and the instrumentation. Biological information tells you what was the quality of your RNA? Was it degraded? What was the overall expression of your housekeeping gene? So it's more of a biological information related to the samples itself, related to the RNA. And so that's distinct from these technical type metrics and visualizations. So I'm going to start going through these metrics one on one. And again, this is going to be slides with a lot of information here. And again, I would just suggest you just kind of think about what I'm saying and try to learn the high level concepts. If you're not able to write everything down, that's no problem. I'm going to send these off as notes to you as well. So let's start talking about some of these metrics, and I want to make sure you understand them. And then after we go through all of these in slide format, I'm going to jump into Rosalind to show you what they look like in the platform. let's get started with imaging quality. So this is one of the very first metrics that NanoString established as a best practice almost 20 years ago now. And so what we have here on the left-hand side, this is a representation of one of the nCounter cartridges. So we have 12 samples in this cartridge. This one lane here represents one sample. So we have 12 lanes in this cartridge, one for each sample. This is here a zoomed in version of one sample lane. And if you're familiar with the technology, you're essentially labeling individual transcripts with these fluorescent barcodes. And then you bind all of those barcodes to this imaging surface. And so to collect your data, you essentially scan this entire imaging surface and simply just count the barcodes that are present that are bound to the surface. So when this is done, the instrument does not scan the entire surface area in one step. It breaks the entire surface area into individual FOVs or fields of view. So in this case, this happens to be a Max or a Pro cartridge. But in order to cover the entire surface area, we need to have 555 individual FOVs that are scanned one at a time. And then at the end, all of these FOVs are stitched back together, and then we can compile our final data and counts of all of our barcodes. So that's kind of this high-level introduction. So why did I say all this? And it's because we have a metric that looks at the performance of this imaging. So the metric is called imaging quality. Its purpose is simply to assess the performance of this imaging in each sample lane. So how well does this imaging perform? How is it calculated? It's the ratio of the FOV counted divided by FOV count. So if you're not familiar with these terms, FOV count is the total number of FOVs that you attempt to scan and collect. In the case above, we are collecting 555 FOVs. So that's the denominator right here in this metric. FOV counted is the number that we actually scanned and collected data from. So in this hypothetical example, in this one sample, we attempted to scan 555 FOVs. In actuality, we scanned 532, and our metric here is 0.956. So you'll get a flag if this value is less than 0.75, indicating there might have been some issue with the scanning or the imaging. Why is this important? Why do we want to look at this? Because looking at this metrics enables us to identify individual samples or entire cartridges that have some sort of imaging or data collection issue. So obviously this is the first step in collecting your data, so it's really important to look at imaging quality first. Now I have all of these pro tips here at the bottom. This is really detailed information. I'm not going to necessarily go into all of this, but I do want to mention this one. Oops, I'm sorry. Let me go back. Didn't mean to scan. This first point here is that the way that I think about the imaging quality metric is essentially it's the percent surface area of the lane that you're successfully scanning. So in this example here, 532 divided by 555, the way I think about this is that in this particular sample, we successfully scanned about 96% of the surface area. So it's kind of an easy way to think about what this metric means. In my experience, typically this metric works, or the imaging works really, really well, or it totally fails. There's very little in the middle ground. So values you probably are seeing is greater than 0.9, maybe 0.95 or higher, or it fails for some reason. Why it might fail is you might have a dust particle on the surface area that's being imaged. There might be a smudge from a fingertip. You're putting the cartridge in the scanner. It might be slightly tilted on one of the axes. So that can impact the scanning as well. Also, if you have too much RNA loaded in the sample, I'll talk about this in the next metric, binding density. But if you actually have too many probes on the surface here, imaging fails because the barcodes overlap. But I'll talk about that in the next slide here. So takeaway from here is if you see a flag for imaging quality, the only thing you can really do is just try to rescan the cartridge. If the root cause of the flag was that there was too many barcodes on the imaging surface, you can't do anything. But if maybe the cartridge was tilted or there was some dirt or something on the imaging surface, simply reload the cartridge and scan it again. So that's the first metric, imaging quality. From there, let's go to binding density. And so binding density is, if we look at this image, it's basically figuring out how many of these barcodes are bound to our imaging surface for a given sample. So we want to look at this because we want to assess if there's too many of these barcodes on any given surface area. It's calculated simply by counting the number of fluorescent spots per square micron that are bound to any given sample lane. So we're just counting these number of spots here and calculating how many there are per square micron on this lane or this imaging surface. So there's certain thresholds here that we have in the software. It depends on the instrument you're using. Those might vary a little bit. But this is important because if these barcodes on this imaging surface, if they're too close to each other, or if there's too many barcodes on the imaging surface, they can lay next to each other, they can start to overlap. And when this happens, the imager can't resolve the barcodes from each other. You start to drop barcodes, drop counts. And if you have too many barcodes, if it's totally swamped, this is something called saturation. The cartridge is saturated with too many probes. And in this case, the imaging might totally fail because there's just so much information, we can't resolve the individual barcodes from each other. Typically, that doesn't happen very often. It's pretty rare. But if you do start to approach saturation, which may happen above these metrics here, then you can get out of the linear range of the assay. So I have a lot of what I call the pro tips on the right-hand side. Again, there's a lot of detail here that I don't want to just read to you, but I want you to notice mainly that this upper limit of binding density threshold, it's conservative. So if you get a value that's slightly above, let's say 2.25, maybe your value is 2.3, 2.4, it doesn't necessarily indicate assay failure. It's more of a caution saying, take a closer look at the sample because you might be approaching saturation, but it's not a true pass-fail criteria. We often get questions about, what impacts binding density. It's a combination of multiple things: how much RNA you're loading, the number of genes in the panel. So if you're looking at, more RNA or more genes, your density is going to be higher. What is the expression level of your genes? If you're looking at high expressors, it's going to increase the binding density more than low expressors were. And the fourth parameter here, or factor, is RNA degradation. So if your RNA is highly degraded, then your binding density is going to be lower than if your RNA was fully intact. I want to point out kind of one other caveat here. It's very rare to see flags for low binding density. In most cases, if you see this, again, which is rare, it doesn't actually mean anything happened that was wrong. I see this most often if you're running a panel. It's a custom panel with very few genes. If we think about, again, the contributing factor to binding density, the number of genes is really important. So if you have a custom panel with, let's say, 20 genes, you're just not going to have many probes bound to the surface area because you're just not counting that many genes. So if you do get a flag for low binding density, it's probably in a small custom panel. At least that's what I've seen in my experience. Okay, so let me continue on here and let's talk about the controls. So if you're familiar with nCounter technology, there are a series of positive and negative controls that are spiked into every sample. And these are really great. This is a wonderful feature about the technology, is having these internal controls. You might not see these in other types of gene expression that you're working with. So the value of these internal positive and negative controls is immense, especially in quality control. So these controls, if you're not familiar, they're synthetic sequences based off of ERCC sequences. I'm not going to go into what those are, but again, they're spiked into every sample and they're contained directly in the probe reagent tubes. So they're added automatically when you set up your reaction. So these are synthetic sequences. They're not going to be interacting or cross-reacting with any of your target transcripts, any of your biological targets. They're pure synthetic sequences. And so in the positive controls case, you have a capture probe, reporter probe, and then a target that's generated through in vitro transcription. It's this linear dilution series from 128 femtomolars down to 0.125. When we start looking at the data, the positive controls are lettered A through F. And so A is the highest concentration, F is the lowest concentration. And we can use this information to essentially assess assay efficiency. So I'll show you what that looks like. On the other hand, we have negative controls. So the way that these are formulated is that you design against these ERCC sequences, a capture probe and a reporter probe, but there's actually no target, no synthetic target present in the assay. So by looking at this information, you can establish your background signal or your limit of detection. So let's talk about how to use these controls in the QC process for your data. First, I want to talk about the positive control linearity. So I think the name kind of gives it away, but we look at these to assess the technical robustness and the linearity of each sample. So the way that this is calculated, and I'm going to show you this in a figure in the next slide. So if this text isn't clicking with you, just bear with me for a minute and I'll show you a visual diagram. But essentially we just compare the concentration of all of these spike-ins, positive A through F, and then what is their raw counts. We do a correlation analysis and calculate that R squared from the Pearson correlation analysis. So the threshold for this flag is that if this R squared is less than 0.95, then we'll flag it as this linearity, or this questionable linearity. So it's important to look at this metric because it allows us to quickly identify certain samples that may have issues with technical performance. It helps us isolate and differentiate between samples that may be problematic at a technical level. Maybe the assay is not working technically versus problems with, let's say, RNA degradation or actual biological issues. So I'll show you what this looks like in a minute. But I do want to mention that typically this linearity, it's independent from your RNA sample itself. So the quantity of RNA, the integrity, the purity of it, if you're measuring your A260 to 280s or 260 230 ratios looking at purity, none of that typically impacts the linearity. We know that this is an enzyme-free reaction, no RT, no polymerase. So any types of contaminants or integrity issues typically don't impact the technical performance of the positive controls. So there may be in some exceptions if you're preparing lysates and you're having too much quantity of salts, but a standard purified RNA sample, that's typically not going to impact the positive controls at all in terms of the linearity. Important thing to note here is the second bullet point is that if you load just a pure water sample or your RNA is completely degraded, the RNA linearity will be excellent. And that's because it's purely looking at the performance of these synthetic controls. So, no metric is perfect. This is not giving you information about your actual RNA sample, but it's giving you information about the performance of the assay at a technical level. And similar to the imaging quality control, I typically see this as working really, really, really well, 0.95 or higher, or it just fails completely. In my experience, I don't think I've ever seen linearity values in like the middle ground, like 0.5. Not to say that doesn't happen, but typically that's not what you're going to see. And then I'll also note here, if you see issues with linearity, there's no real stretch computational strategies you can take to adjust for this. You just kind of have to take the data as it comes. So let me show you what this looks like conceptually. If you want to visualize what linearity looks like, we take the data in the table on the left. We have our 6 positive controls, A through F. We have their concentrations of the spike-ins and then the raw counts. So that's plotted in the figure on the right-hand side. I will note to do this correctly, you need to log2 transform the data. And the also important caveat here is that positive F is not used in this calculation. We'll see that in a couple minutes, but the limit of detection for the assay is established using positive control E. Positive control F has a lower concentration than positive control E, so we're not going to use it in this linearity calculation. So when we calculate this, it's only positive control A through E because technically those are the ones above the limit of detection. So we do this plot, perform our Pearson correlation, calculate that R squared value, and the value of that R squared is the value of our metric. And again, if this value is less than 0.95, then you get a flag for positive control linearity. Let's keep moving on here. Next one I want to talk about is, again, related to positive controls, but now let's talk about positive control A. So this isn't necessarily a metric by itself, and this is going to be kind of my unique take on this. We've added it into the Rosalind platform at the recommendation of the NanoString team, and also I've been teaching this for a decade now, that it's always important to look at just the positive controls themselves. It's not a true metric, but as this example in the middle, this is the positive control values for, I don't know, 10 or 15 samples here. And it's always good just to take a quick look with your eye, scan across to see what you can see. You might easily see if one sample has a really low value of positive control A, like 100, clearly there's something wrong with that sample. So it's always good just to do kind of this reality check, look at the positive controls A quickly with your eye. I noticed down here at the bottom that these are about 2 times higher than the other ones. Maybe that's important, maybe it's not, but at least I'm aware of that as I progress forward in my analysis. So the purpose of looking at positive control A, if I go back to my talking points here on the left, is that allows us to look at the variation of the performance, the technical performance across all samples. So how is it calculated? You know, I put a lot of words here for future reference, but it's simply you just look at the values, the raw counts of positive control A. So that's all it is. So there's no threshold, there's no quantitative assessment here. It's purely quantitative. I want to look across here and see how much variation there is across all my samples. And again, this might help isolate technical issues in the instrumentation or the chemistry versus RNA or maybe RNA degradation types of issues. So on the right-hand side, I give all this detailed information, these pro tips that you can take home and study further after this session is over. I did mention looking at these values to assess technical variation or technical variability. I do want to define what I mean by that. So when we think about the assay itself, there's a lot of steps. So you start in the lab, you set up the hybridization, you pipette your reporter probes, your capture probes, you pipette the RNA itself. You set that up in the thermocycler that runs overnight. You put it in the instrumentation. You have magnetic bead purification, binding of all of your molecules to your cartridge. You have a lot of washing steps. Then there's imaging steps. So there are all these different technical steps in the assay, and each one by itself is a source of technical variation. That's the way all assays work. And so when I talk about the technical variation of the assay, kind of all of these individual steps are reflected in positive control A. So all the variation we see here just reflects this natural variation in the assay encompassing all of these steps that I just described. So what I also want to point out here is that if you see a difference and you say, is this difference, these ones in the 20,000s relative to the ones in 10,000, is that too much of a difference? We actually have an additional metric that we'll talk about in a couple minutes. It's called positive control scaling factor. And that's actually going to do a quantitative analysis telling you if any of these samples by themselves have too much technical variation than we expect to see. So this is just the first pass. What do I see with my eye? Is there anything problematic? And then in the second QC metric that's going to come, we can assess at a quantitative level, is there actually too much technical variation in a sample? And we can show you that in a QC metric. Okay, so that being said, let me continue on our discussion of the positive controls. Let's talk about, the negative controls as well. And let's talk about the limit of detection metric and positive control E. So limit of detection, as the name implies, we look at this because it allows us to assess the sensitivity of the assay in each sample. So it's simply a comparison between two values. So we look at the value of positive control E in a sample and compare that to the limit of detection in a sample. I'm going to show you a visual in my next slide what this looks like. So if you're having trouble understanding, just bear with me for a minute. And the threshold for this QC flag is if positive control E is less than the limit of the detection. So this is important because it allows us to identify any particular samples that might be having some issues with low sensitivity or technical performance. I have my detailed list of kind of my insights that I've written down here for future reference. But what I want to emphasize now is that in my experience, it's very rare to see an issue with the limit of detection in isolation. I don't know if I've ever seen a sample where every piece of QC metric, every QC metric looks great except for positive control E. From what I've seen, positive control E flag is usually triggered by some other issue. So that's just my experience. Your experience might differ, but I wanted to share that for future reference. And when this flag is triggered, typically I see it because the value of positive control E is decreased and falls below the limit of detection. The other scenario would be is if the actual limit of detection increased above positive control E and positive control E did not move. I may have seen that once or twice in the past 10 years, but it's much more rare. Typically this happens because there's something with the positive controls and positive control E just drops below your limit of detection. So that's what I've seen most often. You might have different experiences, but I wanted to share that for your consideration. So let's visualize this in the next figure here. So that's shown here. We have some positive control values in that top table that I've displayed before. Below that, this is the negative controls in the same sample. So this is data for one sample. And what we're doing is we are plotting positive control E. and this is in the log2 scale again. And then we're calculating the limit of detection based off the negative controls. So in this hypothetical sample, we have 8 negative controls. We take the average of the negative controls and add 2 standard deviations, and that's what we define as our limit of detection, and that's this red dotted horizontal line. So if this positive control E is greater than or higher than on the y-axis, it's above this dotted line, then we pass the metric. Sensitivity is good, the assay is performing as expected at a technical level. If this positive control E drops below or underneath this dotted line, then we get a QC flag that's thrown, and that's what's written down here. If this positive control E drops below the limit of detection. Okay, so I think I have maybe 2 or 3 more slides to go through, and then let's jump into Rosalind and get some questions. Next metric, which is brand new, available within Rosalind, not available anywhere else, is this metric we call housekeeping genes greater than 50. So it's a pretty straightforward metric and allows us to assess what is the abundance and the integrity of the RNA in each sample. So if you're familiar with the concept of housekeeping genes, these are historically thought to be these constitutively expressed genes that are going to be kind of robust and consistent across all samples and maybe all tissues. That's not always going to be the case, but the point here is that looking at the expression of housekeeping genes, seeing if they're high or low, gives you an idea of how much RNA there is in your sample and whether or not it's degraded or intact. So there's this simple formula here to calculate this value. So in your sample, we look at all of the housekeeping genes. So let's say in this particular hypothetical panel, we have 20 potential housekeeping genes. So that 20 is going to be in the denominator here. And so we have 20 total potential housekeeping genes in this sample. And then we look at those 20, how many of them have raw counts greater than 50? If it's 10, then our metric here is 10 divided by 20 or 0.5 or 50%. So it's simply the ratio of all potential housekeeping genes in your sample, how many of them have raw counts greater than 50? As a side note here, there's some confusion about, does this housekeepers, is it the housekeepers used in normalization? Is it all housekeepers? But just want to emphasize here that we look at all housekeepers, not only the ones used in the normalization. It's just all housekeepers in a sample. So again, this is important because it allows us to quickly identify any samples where RNA abundance or degradation may be an issue. I have my detailed notes here on the right-hand side. I think the takeaway message for you is that there's this caveat down here at the bottom. And what we see is that this metric works really great if you have maybe one of NanoString's off-the-shelf panels where you have 40 potential housekeepers, 20 potential housekeepers. You have this large set of potential housekeepers. And so this metric works really great in that context. I've seen in smaller custom panels where you might have, let's say, 3 potential housekeepers or 5 potential housekeepers. This metric is a little more variable and a little less predictable, just because what's happening is that if we look at this equation on the left-hand side, the denominator, this total number of housekeepers in the panel, is just so much smaller that if you just change one housekeeping gene either above or below 50, it has a large percentage effect on the metric. So my takeaway for you from this is that this metric works really well with these kind of off-the-shelf panels or a custom panel with a couple dozen or a few dozen housekeeper genes. If you have a very small targeted panel with maybe 3 or only 5 potential housekeepers, this metric is a little more variable and unpredictable. So just keep that in mind as you're looking at your data. Okay, so let's talk about 2 more, and then let's go into Rosalind. So there's 2 that I'm going to show. One is called housekeeping gene scaling factor. The other is positive control scaling factor. And so what these allow you to do is compare, well, let's focus on housekeepers first. It allows us to compare what is the abundance of housekeeping genes used in normalization across all of our samples. And this isn't going to make sense now, but I'm going to show you a spreadsheet of what this is going to look like. But the steps are shown here. So written out in full form, I'm going to show you what this looks like in Excel so it makes more sense. But what this allows you to do is identify, again, any samples that have really, really low housekeeping gene expression that's relative to all the other samples in the data set. So it's a really good way to identify outliers that might happen during the normalization process. So I think that's going to be the takeaway from this message. Again, there's a lot of detailed information here, but it's really a great metric to look at and identify any samples that have really, really low housekeeping gene expression that might impact normalization. The next metric that's related, that's actually calculated the same way, except you're using the positive controls, is positive control scaling factor. It allows us to look at, in your data set, what is the relative performance of the positive controls from sample to sample. The calculations written down here are the same as for the housekeeping gene scaling factor. So the only difference is you're looking at positive controls instead of housekeeping genes. But my takeaways are right here on the right-hand side. And typically what ends up happening is that there might be one sample where the positive controls are just much, much, much lower than another sample. And then this metric is going to pick that up, because it's possible, and I've seen it many times, where a sample might have really low positive controls, but they're still linear. So your positive control linearity metric is not going to capture the sample, but it's going to be picked up in this type of screen here. And again, I can show you what this looks like, but it's a nice complement to the linearity metric to ensure we're looking at both the linearity and then also what is that overall expression of the positive controls relative to the other samples. So again, this might be a little cryptic right now, but I'll show you a spreadsheet to make this a little more clear. All of this is written down for your future reference. And so what I've done is I've kind of detailed here what I would recommend as the QC procedure to analyze your nCounter data. So this is for gene expression. First, I always recommend looking at the raw data. I don't know how many of you are doing this or how popular this is, but just open up the raw data right away. Take a look at it. What do the positive controls look like and the negative controls? What are the raw counts? You start to learn a lot about your data when you can just start looking at things, even before looking at the metrics, because this is a really nice reality check. Again, as I mentioned before, as an example, if you run a water lane, no RNA present in your sample, positive controls and negative controls are going to look great, but your counts for all of your endogenous targets is going to be 1 because there's no RNA. So that's the type of thing you pick up right away with your eye, but it might not be captured directly in a metric. So always look at the raw data first. Then I would recommend going through your QC metrics in this fashion here from top to bottom. What I've worked with our development team to actually organize the output of Rosalind QC to follow this particular path here. So it's, we kind of, whether you realize it or not, we are escorting you and guiding you through this recommended path from left to right of how to interpret and the correct order to interpret these metrics. So there's a lot of intelligence and thought that goes into how things are laid out in the platform. And again, it's to facilitate this logical progression of QC metrics. After that, I recommend looking at the QC visualizations. I don't have a preferred order for these. You can look at them in any order you want. And the fourth aspect here is something that is typically, it's a great kind of hidden secret. I've always said that the meta-analysis feature within Rosalind is one of the most powerful features of the platform, but the least appreciated and understood. And one application, and this is just one minor application, is it can be used to assess, what is the impact of outliers or including or excluding specific samples. So you could set up a meta-analysis comparing your data with an outlier versus without, and then you can see at the gene level, what is that difference? And that can help you decide whether or not you want to include a questionable sample. So meta-analysis is a really great tool for this quality control. It's kind of this hidden secret, but I want to make sure all of you are aware of it. And we'll have a meta-analysis session, I think, in a few weeks, and we can talk about this in more depth. Okay, finally, there is going to be a decision tree coming soon. So the idea of this is to kind of map all of these metrics we've looked at, all of the thresholds, all of the decision points into this diagram you can follow from top to bottom to get a really nice logical progression for your data to make sure you're making the right decisions and you understand the implications of progressing from metric to metric and including or excluding specific samples. So we are finalizing this in the background. We're putting a lot of thought into this to make sure it's robust. So that will be coming soon. Okay, so I'm gonna jump out of here. Looks like we have 10 minutes left to do a quick show within Excel and then the Rosalind platform. So this is some data showing us raw counts in these 5 hypothetical samples. I want to talk about those positive control scaling factor and housekeeping gene scaling factor metrics. So the math is the same. I could be performing the exact same thing in positive controls, but just simply for the sake of time, I'm going to show you what it looks like on the housekeeping genes. So what we're doing is, in this data set, we have 5 samples. We have this large potential set of housekeeping genes for normalization. We have selected that we want to use this set of housekeepers for normalization. So let's assess kind of the relative abundance of these housekeepers in all the samples. So what we do in each sample, so we take this data here, sample 1, our housekeeper is selected for normalization. What is the average? In this case, we use the geometric mean. So we calculate this geometric mean. Here is the geometric mean of all of these genes in sample 1, sample 2, sample 3, sample 4, sample 5. Then we simply take a look at all of these. We take the global average. So we're going to take just the global average of this, the global geometric mean of all these values. That's represented here. Finally, to make our factors, we just do a ratio. It's simply this value versus this value. So this 1.26 is calculated by this global mean versus sample number 1. This 1.37 is the ratio. It's the difference between this mean and sample number 2. And so this is the ratio between the global mean and sample number 3. So the way to interpret this is that what the value means is that it's the distance between the expression, the average expression of housekeepers in a given sample versus the average of the whole experiment. So as an example, this is 1. So it's basically telling us, within rounding error, that the expression of housekeepers in sample number 3 to 11 is basically the same as the average expression across all the sample. In this particular example, sample number 1, it's telling us that the average expression of housekeepers in this sample is basically 1.26 times less than the global geo mean. So it's a great way to identify, when we look across all of these 5 samples, what is the distance of the expression of the housekeepers of, let's say, sample 5 versus the average, or sample 1 versus the average. So that's how you kind of interpret it within the platform. So let's do a quick review in Rosalind, and then I'll answer some questions. I got logged out here. Let me log back in, and I'll show you what the QC looks like in the platform. So I have a data set ready to go. I'm going to find our QC table, open it up, show you what the exploration looks like. Okay, so let me just jump to our quality control results. And so when you come to your experiments, all the QC metrics that I just described in depth are represented here in this particular table. In this demo data set I chose some samples that had some QC issues. If you log in and you see this blue banner at the top when you come to your QC page, then that indicates you might have some QC flags. So what those look like is different colors. So this red box indicate this particular metric in a particular sample was flagged. Yellow indicates it's right on the threshold itself, so it's right on the border. But the way that I recommend working through this is, in the first column you have your sample names, then look at how well the imaging performed. So in this case, it was really high quality across the board, no issues with imaging. Then let's look at the binding density. Was there too much RNA loaded into the sample? Are we even close to saturation? The answer is no. This all looks great. These are all within our preferred range of binding density, so there's no linearity issues, there's no saturation issues, so we can progress to the next step. Now let's look at the technical performance of the assay itself. Let's look at the positive control linearity. How linear and how robust are these positive controls across all of our samples? In this data set, it's excellent. There's no concern with linearity or any of the particular robust performance of the positive controls in this particular metric. Then we can look at the scaling factor. So this metric is telling us, for a particular sample, what is kind of the average expression of the positive controls relative to the average of the whole experiment. So the average expression of the positive controls in this sample is 1, so it's basically at the average of the whole experiment. So these are all really close to 1, which is what we want to see. It indicates that all of these samples are really consistent in terms of their positive control expression. Then we can look at positive control A. Again, this is going to be used to look at, what is that natural variation across the data set? This ranges from about 30,000 to, yeah, they're all in the 30,000s, maybe as low as 29,000. Not too much variation that I can see by my eye. As kind of a reality check here, we can again look back at our positive control scaling factor. If any of these values were flagged, that would indicate there's probably too much variation in the positive controls, and we should take a look at that. Next, we can look at our limit of detection. So this is the value. We took it in this particular sample. We take the average of the negative controls, add 2 standard deviations, and then we actually compare this value to positive control E. So if this value is less than the limit of detection, then you will get a flag here. In this case, this is well above. So all these positive control E's are much, much higher than our limit of detections, which is the column to the left. So clearly there's no issues with sensitivity in this data set. It's performing really, really well. Finally, we can come to our housekeeping genes above 50. In this case, there might be a couple samples here where we're flagged below 75%, which is our threshold. So maybe you want to come in here and look at these samples. What is the expression of the housekeeper genes? Is it robust? Are we getting good normalization? Finally, we have housekeeping gene scaling factor. And this is, again, comparing the relative expression, the relative average of housekeepers in one sample versus the average of all other samples in the experiment. So there's a lot more we could go into, and I know I'm running low on time here. We have some visualizations we can explore. But let me show you what I do recommend doing in addition, and then I'll have some questions, is always go to your file management section and then come down here and go to your count matrices and download this all probes with raw counts file, and always do a reality check. Look into this file, and what you'll see is all of your raw data in a table like this. You see all of the raw counts for the positive controls, negative controls. You can easily spot just quickly visually, are there any issues with the negative controls? These look very consistent. Are there issues with the positive controls? And then linking back to my previous master classes, we talked about custom pruning and custom normalization. We have these 2 columns which have a true false flag indicating, was the gene pruned from the data set, yes or no? And was the gene used in normalization, yes or no? So always, always, always look at this raw data file. It allows you to quickly identify if there's any issues with controls or even in the raw counts themselves. So there's 2 minutes left. I didn't leave much time for questions, but I'm happy to stay on and answer any questions people may have.
Thank you so much, Ryan. Yeah, I think there were a couple questions. So the first one was, what if your experiment doesn't have the positive control scaling factor?
If this is a question about having it within Rosalind, it's most likely that this was an older data set. So this new positive control scaling factor was a pretty recent addition to the platform. So if you don't have it in your analysis in Rosalind, you simply need to re-upload your RCC files and regenerate the analysis.
Awesome, perfect. Let's see, one more question. So I know you said that we would have the decision tree soon, but I guess the question is, how do I establish what are good thresholds for some of these metrics?
Yeah, so the thresholds, so 2 things. The thresholds, if these metrics did exceed a threshold, it would be flagged directly in the Rosalind platform. So you'll see it here visually. If I go back to my slides, which we'll be sharing, then all of the thresholds are written in the slides. And then we also have Rosalind Academy, where we have information about how QC metrics are calculated, how they're interpreted, and what the thresholds are there. So there's 3 places. In the platform here, you'll see the flag. You can refer to the slides, and you can also refer to Rosalind Academy.
Okay, awesome. One more question. What QC plots does Rosalind provide?
Sure. Yeah, those are shown here on the left-hand side, and apologies for not having enough time to go through these, but we have a nice plot that looks at the correlation of all your different samples. So this is a Pearson correlation, allowing you to identify if your different biological replicates have a really strong correlation with each other and whether or not they cluster with each other. So this is a really nice kind of biological QC plot. We have a violin plot, which can be quite interesting. It's showing you the distribution of your counts. So this would allow you to identify any outliers pretty quickly. If there was a very marked shift of one sample relative to the other ones, then clearly something might be wrong or different about that sample. So the violin plot can be a really nice quick assessment. We have visualizations for all of these positive and negative controls and housekeepers that I just talked about in the slides. So you can look at what is the expression of the positive controls. We have a view of the stacked percentage graph for the positive controls and negative controls. We have plots for housekeeping genes as well. And I will point out that you can download these as well. So you'll see there's these download buttons. So you can download SVGs or PNGs. In some cases, you can also download the CSVs to get the data behind the plots. We have this variance of mean plots, which is showing you essentially the performance of your housekeeper genes. You can certainly answer more questions about this, but it's just basically showing you that the genes used in normalization, shown here in blue, have quite low variance relative to the other potential housekeepers shown in orange. And we also have an MDS plot. So this is a multidimensional scaling plot. You're de-dimensionalizing your data set, allowing you to look at the relationships and the similarity between your samples. Very similar in principle to PCA, or principal component analysis. And you can layer on top your different attributes. In this case, we have different time points and different treatments. So that's a quick overview, Kiki.
Thank you so much.